All right, so here's our DNA structure notes. And so if you haven't opened them yet, go to Classroom. You're going to click on Unit 5 and go scroll down till you find the DNA structure notes and open up your Cornell notes. Remember, you got to open up the one that has your name on it. So you want to open up the entire assignment and it should be over on the right, okay? So you should have this document open. Remember, you can also write this down on a piece of paper and then type it in later. So you want to make sure your name is up here. It says your name and then DNA structure notes. All right, so let's go ahead and get started. All right, so when we're looking at the DNA, we're going to talk about how it's structured and why it's so important. So we talked about mitosis and that it's replicated. Now we really want to know, how is it replicated? What does it do? Okay, so our essential question here for these notes is, what is the structure of DNA and how does it replicate itself? All right, so DNA actually stands for, it's a very long name and this is why we call it DNA, is deoxyribonucleic acid. So it really tells us basically this is all the stuff that it actually contains, okay? And DNA was actually thought that it was flat and we weren't sure how it kind of fit in there, but James Watson and Francis Crick actually came up with a new model. They built it, and there's many sculptures and stuff built after them, but they actually found that it was the shape of what we call a double helix. And why do we call that? Well, you can see there's two sides to it, and then it's twisted like this. And this is how it's kind of shoved in small places because there's so much of it. So Watson and Crick were actually the first to actually come up with what shape it would actually be to fit into all of our tiny little cells. All right, there's a couple facts I want you to know about DNA. DNA actually codes for your genes or your traits. What the heck are those? Those are your eye color, your hair color, your height, your skin color, the shape of your nose, what your lips look like, your eyes look like, okay? All of those things are coded within our genes, and those are all called traits. And they're made of repeating what we call subunits called nucleotides, okay? So the nucleotides, we're going to go in more detail, are these here. So each side, you can see how they're attached. And we're going to get into more of this, but this is a 3D model of what DNA actually looks like. There's 3 parts to DNA. Okay, it's what is a nucleotide, right? Remember when we talked about monomers and polymers, when we talked about nucleic acids were the polymer and the monomer was a nucleotide, so one part of it. And so it has 3 parts. It has what we call a phosphate, which is this ball-looking thing group, and you'll learn more in chemistry what that is. Deoxyribose, which is a form of sugar, which is here. And then a base, which is the nitrogenous base. And these are different, but this phosphate and this sugar are always the same in every single nucleotide. And we're going to learn about the different bases, A, T, G, and C in a little bit, but that's what makes the things different, and that's what codes is actually these nitrogenous bases, because the phosphate and the deoxyribose sugar are all the same. All right, so remember, I just told you that we have the deoxyribose and the phosphate, and those are all the same, and we call those the backbone. We'll hear about that in a minute because those are the same, but what's different is our nitrogenous bases in the center. So we have 4 of them. We have one that's called adenine, A, which is represented by the green here. We have guanine, G, which is represented by the purple, thymine, T, which is represented by the red, and cytosine, C, which is represented by this green. So you can see only certain ones pair with each other. So A always pairs with T, so we can look here. So you see adenine, and then you see thymine on the other side. Adenine, thymine, adenine, thymine. They do only pair with each other. And then guanine, G, which is our purple, always pairs with cytosine, and they go there. So G never pairs with A or T, and C never pairs with A or T. A always pairs with T and G with C. So that's one of the memorization things you're going to have to kind of memorize in your brain. Okay, and the sides of the DNA ladder, we call, those are our sugar and phosphates are these. So the blue, light blue, and dark blue, those are your sugar and phosphates. And they're held together by what we call hydrogen bonds. And you'll learn how strong those are once you get into chemistry. Just know they are held together by hydrogen bonds. What does that mean? It's because the atom hydrogen is in there and it's bonded to something else. So you can see how the ladders, the rungs of those ladders are bonded together here. Okay. All right, so when we talk about the base pair, we talk about how they pair. So remember, I said A goes with T, T goes with A, G goes with C, and C goes with G, right? So if I have on one side ATA and then a TCA, you can see that A, the other side, would be a T, and a T, the other side would be an A, and A, the other side would be a T. So I want you to take a minute. I want you to try, just on a piece of paper, or even on your notes, write out what you think the other side would be. All right, so hopefully you gave it a try, because you will have to do that on a few exams. So hopefully you got T-A-T, and then T goes with A, C goes with G, A goes with T, T goes with A, G goes with C, C goes with G, and then 3 G's go with 3 C's. Okay. All right, and this is kind of what it looks like, right? So we have a DNA. So this is just a small section of the DNA. You have the nucleotide, which has our phosphate group, okay, our deoxyribose sugar, and then our nitrogen base here, okay? So how does this code work? Well, the combinations of A, T, G, and C determines what traits you might have. For example, if you have something that has a CAT and a CAT, that would say, hey, you got purple hair. But if I turn it backwards, TAC, TAC, that might say you have yellow hair. So the order that they come in basically says what your traits will be. And there's millions and millions of different combinations that can be there. Okay. And think of the bases of DNA like letters. Just think of them like letters. I know we talk of that they are chemistry. You're right. But this is kind of a good concept to overthink everything, especially as we get into protein synthesis after this. Remember, these are our letters, okay? And these letters, so these bases, form words. And those words form sentences, correct? And there's endless combinations that you can do. And if you change the words, or you change the letters around, you're going to change the words, which is going to change the entire sentence. So it's kind of thinking that like these bases, okay, form our genes, which are words, which then form our entire traits, which are their sentences. So as we combine them differently, that's when we can get completely different traits or combinations, okay? All right, so now that we kind of know how it's put together, let's talk a little bit how it's replicated. Remember when we talked about the cell cycle, we talked about the synthesis and how DNA is copied. Well, we're going to start really getting into how it's copied. So the process by which DNA makes a copy of itself is called cell division. We learned that, right? So actually, instead of just taking it and copying it, we don't have a copy machine inside our body. So what it does is you have your old strand. It actually comes through, and there's different things, different enzymes you'll learn, in AP biology or up in college, that there's things that come in and basically they do what they call unzip it. So they separate those bases, okay? And then what the body does is it brings other enzymes in and it starts to pair up again. So you know that if this was an A and this might be a T, right? So now we have an A on this side and T on this side. It's going to separate. Then it's going to come in and match the bases again. So then in the end, we have 2 copies now of our DNA. But each copy has one old piece and then has the new piece it just made. So it unzips it, makes a new side of the other zipper, and now we have 2 copies. And we call that semi-conservative. basically means half of the old strand is saved. So it doesn't just copy. There is no photocopy. There's no Xerox machine inside our body that we can just put it on and it copies it and goes. So to make it easier and faster, it actually unzips whatever it needs to copy and then just makes a new strand to match it with the bases. All right, so that was our DNA structure notes. So go ahead and process through your notes. You're going to highlight any important notes. You should make connections to any of your notes in the left-hand column. Write any questions you may have in the left-hand column, and then use your highlights, connections, and questions for your summary. And before you turn it in, please make sure you have highlights on there and that you've written your 3 to 5 sentence summary. If you have any questions, please let me